Dear student, today we will study Drosophila melanogaster. Drosophila melanogaster is also known as fruit fly. This is used as a genetic tool to study linkage and crossing over, sex-linked inheritance, and also used to create chromosome map. Drosophila was first studied by Thomas Hunt Morgan in 1910. In 1933, he was awarded with Nobel Prize for pioneering work in genetics for gene theory of inheritance, and he is considered as father of experimental genetics. Why it is used as genetic tool? Let's study what kind of advantages it offers in the study of genetics. It has got a short lifespan of about 14 to 15 days at 20 degrees Celsius, and they are easy and inexpensive to breed. Like you can grow them on mashed banana. The breeding could be easily controlled in laboratory. In a single mating, it produces large number of colonies or progenies, so you can study what is inherited in the next generation. Easy to handle its genetic information, as only there are 4 pairs of chromosome which are present. Sexes in Drosophila are easily distinguishable. That is, the male is smaller, darker, and has blunt abdomen, whereas female is large, light, and narrow abdomen. Linkage and crossing over. Let's see how it was used. So, what is linkage? The genes located on the same chromosome will tend to inherit it together during meiosis to retain parental combination. This is called linkage, because when the genes are located very close to each other, then there are more chances that they will be together inheriting in the next generation. So all those genes which are located on one chromosome, they are all linked. Okay, so all the genes that are located on the same chromosome will behave like a single unit, so they form one linkage group. So total number of linkage group in an organism correspond to the haploid number of chromosome. Like garden pea, 7 linkage group are there. In human being, 23 linkage group are there. In Drosophila, 4 linkage group are there. Now you need to understand this. Let's consider A and B are the genes which are located on same chromosome. So this is how they are located, okay, in the duplicated form. So when no crossing over occurs, they form 2 types of gametes. Okay, so they are forming 2 different types of gamete, that is capital AB, small ab. Okay, now when the crossing over occurs during meiosis, 4 different gametes could be formed. What are the 4 possibility? 2 are these and 2 are new combinations, that are capital AB, A and capital B. Out of these 4, the 2 are parental and 2 are recombinants. So there is 50% parental type, 50% recombinant. In case of linkage, all gametes are of parental type, whereas if there is less linkage, the parental type gamete possibility become less than 100%, because you will see new combinations also. At this time, there are more chances of crossing over. Now what is crossing over? It is the process of exchange of genetic material between non-sister chromatid of 2 homologous chromosomes. The closely situated genes show strong linkage, while those widely separated have more tendency of crossing over. You also need to understand what is mutant and wild type. An individual having the normal phenotype that is generally found in a natural population of an organism is called wild type. It is represented by WT or with positive sign. An individual having phenotype that differs from normal phenotype is called mutant, and it is represented by capital M and negative sign. Okay, so during the experiment they use different signs and symbols. Actually they were used by Morgan. Now let's study the characters in Drosophila. So here I am taking 3 characters, wild type and mutant, eye color, body color and wing shape. If you look at wild type, eye color is red, gray body color and vestigial wing. And mutant, the color of eye is white, black body and long wing. Now here capital represent the dominant and the other one recessive. Here the dominant is red and white is actually not mutant, we can say, but this is actually come under the sex link inheritance, so we'll be discussing later on. Right now the cross is done between 2, that is wild type and mutant. So just look at the wild type character. So gray and vestigial are wild type, whereas black and long are mutant. So male and female crosses is done, they are parents, and then gametes are formed. So only one type of gamete is possible, both the places, and the fusion occurs, and in the F1 progeny you'll get this combination, that is all gray and long. Then next test cross is done. In the test cross, what is done? F1 male is crossed with homozygous recessive. So this is test cross, homozygous recessive. Let's look at back. The character homozygous recessive is black and vestigial. So this was chosen, so test cross was performed. So if you look at the gamete here, the possibility of 4 different gametes are there, 4 different combinations are there, and here only one type of gamete is there, that is a small b, that is black and vestigial. Now this black and vestigial is crossed with all 4 combinations, so you'll get 4 different combinations. Now when this experiment was done by Morgan, he could only see that when he obtained the progenies in F2, he could only see 2 different types of colonies, that is gray vestigial and black long. But these were expected, but he was unable to observe gray long and black vestigial. So if you see gray vestigial, black long, gray vestigial, black long, see gray vestigial, black long. So actually these are the grandparent combination. Who are the grandparent of F2? The parents are F1 and their parents are grandparents. So you will see here grandparent combination. So this indicates that capital B, small b, that is, and small b, capital V are completely linked gene, and recombinant gametes are not produced during meiosis. Therefore, genes for gray body and vestigial wing in male Drosophila exhibit complete linkage. There is no recombinants which are produced, as the F2 show phenotype of grandparents in 1:1 ratio. Now let's discuss what is incomplete linkage. So we will study in the same example in which some percentage of recombinants are obtained. Recombinants means which are possible with the combination of 2 different genes, and combination is only possible when crossing over occurs in the meiosis or during the meiosis. The chances of separation of linked gene depend on the distance between them. If the distance between 2 genes is less, then more possibility of linkage. If it is more, then more possibility of crossing over. So similar cross is there, only here reciprocal cross we are performing. This is same as the last cross. F1 will get gray long, but here reciprocal cross is performed. Over here the F1 instead of F1 male, F1 female is taken, and this is crossed with homozygous or recessive, you can say male. So the possibilities are here 4 types of gametes will be formed, possibility of 1 type of gamete only, and then fusion occurs. So when the Morgan did this cross, he found that the gray long and black vestigial, which are the which are the, you can say, recombinants. Okay, whereas gray vestigial and black long, okay, they are the parental grandparental combination. So if you calculate their recombination frequency, we'll come to the point what is recombination frequency. First try to understand here. He was able to calculate and he found that it is 41.5%, 41.5, whereas in case of recombinant it is 8.5 and 8.5. so when it is combined, then 8.5 plus 8.5, which stand for 17%, they are recombinant, whereas the parental combination are 83%. So more of the parental type means more chances of linkages there between these genes and less chances of recombinant or less chances of crossing over is there. So such kind of linkage where there is no complete linkage, partial linkage is seen, but it is more than 50%, then we say this is called as incomplete linkage. So as the recombination frequency is less than 50%, sorry, so the genes are said to be partially linked and this is called incomplete linkage. Now let's study the crossing over here. Crossing over is the physical process in which there is a reciprocal exchange of corresponding part of chromatid between homologous chromosome, which result into new combination. Like this is homologous chromosome, you can study A, B, C, D. Okay, so 1, 2 homologous chromosomes are there. When crossing over occurs, so they'll exchange the segment, okay, and new combinations are possible. So half of the arm of chromosome will have parental combination. So when they form the gamete, out of one half parental and half recombinant, and half parental and half recombinant is possible. Mendel, when he was doing the studies with pea plant, he missed the linkage. He never said that the gene are linked, but he said that gene are independently assorting. Because luckily it happened that 7 traits which he selected in pea plant were located on different chromosomes, so there were no interaction that could be observed between genes. So he said that they are independently assorting. Okay, so but now this is again a kind of deviation from Mendelism, because here 2 genes, they are showing interaction in the form of linkage. Now, how to calculate the recombination percentage? You should know that genes are arranged on the chromosome in the linear fashion, and the frequency of crossing over between 2 genes is directly proportional to the distance. So more far are the genes, more are the chances of recombination. 1% crossing over between 2 linked genes represent 1 unit of map distance between them. So 1 map unit distance is equal to 1% recombination, which is equal to 1 centimorgan. This is the unit for the gene mapping that we use. Now how to calculate recombination frequency? Now when this cross is done and number of progenies that we are calculating either in F1 or F2, F2 especially when test cross is done, how many number of recombinant offspring are there, and what are the total number of recombinants. So out of that, how many recombinants are there, they are taken and they are multiplied by 100. So whatever number comes, that is your recombination frequency. You just make note of it that whenever these experiments are conducted, they are not conducted with one set, they are conducted with number of sets. Okay, so you'll get number of progeny maybe in thousands. Okay, and after that this is done. The phenomena of separation of linked gene to form new combination of parental gene is known as recombination of gene, and offspring with new combination of characters are called recombinants. By determining the percentage of recombination, we can determine chromosome map or linkage map or genetic map. Now linkage map. Why it is known as linkage map? Because here you can make out that since the genes are located on the chromosome, so it is known as chromosome map, and these genes are linked with each other, so it is known as linkage map. And since we are talking about various genes, so it is also known as genetic map, because you get the sequence of gene on the chromosome. So chromosome map is a graphic representation of position of gene of a linked group in a linear fashion on chromosome. Morgan and Stutman have prepared chromosome map of Drosophila, man and maize. Chromosome map provide exact location sequence of gene in a linked group and indicate strength of linkage and chances of crossing over. So once you get the sequence of gene, you will come to know which is far, which is near, to among which one the linkage probability is more, among which one the crossing over probability is more. So here is one example to explain you. Now recombination frequency between black body and vestigial wing is 18%. Recombination frequency between black body and cinnabar eye is 9%, and recombination frequency between vestigial wing and cinnabar is 9.5%. If you look at the distance in map unit, it will be 18, 9 and 9.5 map unit. So if we try to construct the map on the on the chromosome, so first you'll plot the distance which is far, so between black and vestigial 18 map unit, and then cinnabar distance between two could be plotted between black and cinnabar is 9, and cinnabar and vestigial is 9.5. So you will get the sequence that sequence of gene is black, cinnabar and then vestigial. So there is more probability of linkage between black and cinnabar and less between black and vestigial. So this is how you can make out from the chromosome map. You can make out a lot of information. Even the mutation is studied. Now what is sex-linked inheritance in Drosophila? While studying Drosophila, Morgan observed single white eye male among red eye female. So red eye is the normal color of eye, whereas white eye is the mutant. Later on, he identified that white eye trait is the sex-linked inheritance. So for that, he performed 3 crosses in order to reach to the conclusion that it is sex-linked inheritance. So first cross is done between the homozygous white and homozygous red, where red eye female is crossed with the white eye. Now since this is since we are studying the sex-linked inheritance, so the cross will be represented as X and Y form. We have already done that. The female are homozygous XX and male are heterozygous for the chromosome. Okay. But here I'm talking in terms of character, red eye and white eye. So red eye and white eye homozygous characters. So male and female are crossed. Then in the F1 you'll get what? That all the male and females are red eye. So here you can see all the male and female are red eye. Then in the second cross, F1 male is crossed with the F1 female is crossed with the red eye male. So when it is done, you will see that red and white. Okay, so in the male population, 2 different eye color is seen. So 50% population white, 50% population red, whereas 100% red in the female population. You will see all female are red, male are 50% red and 50% white. Then he performed the third cross, in which F1 red eye female, F1 is which one? This one, okay, is crossed with the white eye male. Okay, now let's check the possibility of result. So here when this is done, you will see that in the female, okay, one is red eye, one is white eye. In the male, one is red eye, one is white eye. Okay, so total population 50% are male, 50% are female. Out of 50%, you can say 50% male are of red eye, 50 of white eye. Similarly, female 50 are red eye, 50 are white eye. Now, since the color of eye is associated with X chromosome, so it is called sex-linked trait. So this is all in Drosophila melanogaster. Thank you.